Thank you. My apologies, I wasn't able to join you in person. Let me start with a personal confession. I'm not brave nor talented enough to build robots myself. So instead, over the years, I've looked for help from two groups, algorithms and people. I'll start with algorithmic help. In my group, we try to create search methods good at navigating robospace, the space of all possible robots. Despite the breathtaking diversity of living forms, evolution has explored only a tiny subset of morphospace, the space of all possible organisms, over the past 3.5 billion years. Over the past 80 years, since the Second World War, we, the robotics community, have explored only a tiny fraction of robospace. What I want to explore with you today is what else is out there. Today, I'd like to take you on a tour to parts of robospace that my colleagues, students, and I have found that are inhabited by particularly non-intuitive machines. That is, machines with form and function very different from any found in nature. Before we begin our tour, a few words about our mode of travel. In all the work I'll show you today, some kind of evolutionary algorithm searches the space of simulated robots as best it can. In this cartoon, each point represents a machine that is more or less competent at whatever we want it to do. It can do more or less things on the vertical axis, and it is more or less embodied, represented by the move from the front of this cube towards the back. Each branching trajectory here is meant to represent an individual trial of this evolutionary algorithm. In any one trial, we typically begin with a population of random simulated machines, delete those machines that perform poorly, make randomly modified copies of the survivors, and repeat this process until we run out of supercomputing time or until our evolutionary algorithm finds a machine in silico that's sufficiently interesting to warrant the extra effort of building it in reality. If we do find such an interesting machine, we try and invite, challenge, or beg our robotics colleagues, many of whom I know are there today, to try and build a physical copy of what our evolutionary algorithms find. Often, though, we fail to cross the sim to real gap. This failure is not because of lack of talent or effort on the part of our colleagues, many of whom are there today. The usual reason we fail is because the physical robot faces a very different world from that encountered by its simulated counterpart. This is true, of course, of robotics in general. Our machines are still not that good at grappling with external change. They're getting better, but I would argue that their difficulty in grappling with external change is because our robots are capable of little internal change. Organisms, on the other hand, grapple with internal change from the moment they're born to the moment they die. There are internal adaptive processes that unfold over time scales from the picosecond to decades or even centuries, depending on the species. And there are internal changes that unfold over the subcellular scale all the way up to the whole body. This inner flux occurs not only across spatial and temporal scales, it also occurs in both the body and the brain. Appropriately, the Scottish philosopher David Hume articulated this particularly well. For those of you that are physically present at the conference, I believe this statue of Hume is just a few hundred meters from where you're sitting. In the 1700s, Hume looked inside himself, looking for that unchanging kernel of self that René Descartes a century earlier had purportedly found. But Hume couldn't find this unchanging self. All he could find were internal changes and feelings that were in a perpetual flux. They were flickering across his mind's eye at a rapid pace. There was no constancy in what he saw inside. In a way, organisms pre-train themselves. They adapt to internal change and have to during development, injury, regeneration, and so on. And by adapting to this, constantly adapting to this internal change, they prepare themselves to deal with external change. In fact, one of the advances in the soft robotics community that I'm most excited about is that we're getting better and better collectively as a community at creating robots that change internally in increasingly interesting ways. Rigid robots are incapable of internal change. They're made of inert metal or plastics. Their bodies can change, however, it's just simply not under their control. They can be damaged by the external environment. This means that the only adaptive path open to them is to adapt their control policies. Soft robots, thanks to many of you, have richer inner lives than their rigid cousins. One feature of internal adaptation available to soft robots is shape change. In collaboration with the Kramer-Bottiglio lab at Yale, we demonstrated how injured soft robots have two options available to them. The first is to adapt their control policy, like you see here, and like rigid robots. We found in this particular experiment that if this four-legged robot was injured in these 9 different ways and was only allowed to adapt its control policy, not change its shape, it was able to recover only a limited amount of functionality post-damage. However, the second option available to these soft robots is to change their shape and just keep using their pre-damage control policy as is. What we found is that, again, in the 8 injury scenarios we explored here, what you're going to see in a moment is one of these particularly grievous injuries. We're going to cut all 4 legs off this robot in a moment, and it's going to search for a way to change its body to recover. And it finds a particularly advantageous way to recover, which is to, in essence, regenerate the lost 4 legs. What we found is in these 8 damage scenarios, most of the time, shape change was able to recover more functionality after damage than policy adaptation was. Sometimes shape-changing robots, like the one you see here, comes up with a particularly non-intuitive way to recover. Remember my promise that we would visit some particularly non-intuitive corners of RoboSpace today. This is one of them. This particular robot discovered not how to regenerate the lost 2 legs, but instead to retract the surviving 2 legs and revert back to a peristaltic form of locomotion. Another type of internal change is change over multiple timescales. Here you see a soft robot that oscillates rapidly in order to move, but changes its overall shape more slowly. You'll notice that the individual voxels that make up this robot, some of these voxels are increasing in volume, some of these voxels are decreasing in volume slowly to achieve shape change. These 2 forms of internal change at 2 different timescales make this robot more adaptive in a particular way. It discovers a rare behavior, rolling, accidentally during its lifetime as it changes shape. In this particular simulation environment, rolling is faster than walking, but it's a needle in the haystack behavior. It's very difficult for a given robot to discover this behavior. That robot's descendants change shape in a way to acquire and hold on to that needle in the haystack behavior for longer. Here you see the ancestor, which clearly doesn't roll, doesn't even move. That ancestor evolved this descendant, which walks well but slowly. This robot produces this descendant, which changes shape and discovers rolling late in its lifetime. Its descendant evolves to change shape in a different way so that it discovers rolling earlier and maintains it for longer. And it, in turn, evolves a descendant that rolls throughout its lifetime. This is an embodied form of the Baldwin effect, discovering behaviors through development or learning, which induces evolutionary selection pressure in later generations to genetically assimilate that behavior. Robots can also stack internal changes spatially rather than just temporally. Together with the Kramer-Bottiglio lab at Yale and the Levin lab at Tufts, we created in silico fractal robots. These robots exhibit self-similar structure and self-similar function at different spatial scales. You can see the self-similar structure here. We've evolved a small robot. We stack that robot together in the middle panel in the way that we built the original robot out of voxels. And then we do that again to produce the robot on the right. It turns out that if you find the right building block, you get not just this self-similar structure, but also self-similar function. All three robots here are moving in more or less the same way relative to their total body size. Future fractal robots like this could be of practical utility. If you build the right fractal building blocks and you discover self-similar structures that confer self-similar function, that could allow you to rapidly create in the field differently sized physical robots that act in the same way. I'll just pause here for a moment to point out that most of the simulation work that you're seeing here today was carried out in Voxcraft. This is a GPU port of VoxCAD. VoxCAD was originally developed by Jonathan Hiller and Hod Lipson back in 2014. The Voxcraft simulation package that you see here was developed by my students David Matthews, Sita Liu, and Sam Kriegman. If internal change indeed pre-trains robots to deal with external change, it should be possible for internally plastic robots to cross the sim-to-real gap better than robots that have no inner flux, have no inner rich life. Anecdotally, at least, I found over the years in working with my colleagues on the sim-to-real problem, it's actually harder to get rigid robots to cross the gap, which you see in the top row, than it is to get soft robots to cross the gap, like you see in the middle row and the bottom left. Of course, it depends on which particular soft robot we're talking about. In general, soft robots have complex dynamics that are hard to port from silico to reality. But again, with the right soft design, sim-to-real is, at least in my personal opinion, not as hard as you might originally think. I'm going to talk in a moment about biological robots, which are shown in the bottom row, where we've adapted this process to create living robots completely out of living tissue. They also seem to cross the gap surprisingly easily. But before I talk about biological robots, I want to digress for a moment and talk about sim-to-real of metamaterials, which is what you see in the bottom right. Many of you here today already build some or all of your robots out of metamaterials. For those of you that don't, metamaterials are artificial materials with tunable bulk properties. In a newly funded NSF project with the Kramer-Bottiglio lab and Corey O'Hearn, also at Yale, and Mark Shattuck at the City University of New York, we are now evolving granular metamaterials. Granular metamaterials, as the name implies, are metamaterials made from small grains or particles. As you can see here, parts of these materials can amplify or suppress the amplitude of an oscillating force, which in these three examples is being supplied at left. We're working on sim-to-real for these materials in 1D. We've moved on to exploring these materials, at least in silico, in 2D, like you see here. Turns out that these 2D granular metamaterials have some interesting properties. If you choose two of the grains, the green and the blue one at bottom, as two input ports, where we can supply vibrations to neither one or both of these two input ports, and if we observe vibrations, if any, arriving at a red output port, we can stiffen some grains, shown dark gray here, and we can soften other grains, shown in light gray here, to produce a computational metamaterial. In this case, we have found a particular metamaterial that acts as an AND gate. where we represent zeros as zero amplitude input vibrations, and we represent 1 as non-zero amplitude input vibrations. And if we combine these zeros and ones expressed as vibrations to the two input ports, we can observe what vibrations appear at the output port. And by choosing which grains are soft and which are stiff, we can get this material to act like an AND gate. Of course, it's not us deciding which grains to soften or stiffen. We wrap this metamaterial simulator in an evolutionary algorithm and evolve populations of metamaterials to exhibit increasing ANDness. Our fitness function rewards materials that act increasingly like an AND gate. We can then expand that evolutionary algorithm into a multi-objective optimization method where every metamaterial in the evolving population is assigned 2 numbers, how well it behaves as an AND gate when supplied with low frequency signals, and how well it operates as an exclusive OR gate if we supply input vibrations at high frequency. What ends up happening, as you see in the bottom left panel, is you get this moving Pareto front of solutions represented by the blue dots that are increasingly good as acting like AND and OR gates. I showed you the AND gate at the top left part of this Pareto front. Here's a different material from the same population with a different different configuration of stiff and soft particles that acts like an exclusive OR gate at high frequency. What I find particularly interesting about this is what happens at the knee or the center of this Pareto front. So if we take the blue point at the center of this front and we multiplex or combine the low and high frequencies, we are simultaneously supplying zeros and ones at different frequencies, and we can observe those different frequencies, if any, arriving at the red output port. And if we read off low frequency and read off high frequency, we can get materials in which that red particle is acting as an AND and XOR gate. This is a form of computational superposition, which does not rely on quantum effects. We're getting one part of the material to compute 2 different functions in the same place at the same time. This is the second non-intuitive corner of robospace, or at least metamaterial space, that I wanted to show you today. The third and final corner of robospace that I'd like to take you to is, to put it mildly, particularly non-intuitive. In 2013, Doug Blackiston, a staff scientist in the Levin lab at Tufts, surgically implanted eye precursor cells into a tadpole tail. Not only did this not kill the tadpole, but during development, the cells and spinal cord wired up to each other, establishing a handshake protocol. And this resulted at the end of development in an adult frog that had an eye on its back and could use that eye to guide behavior. This led us, when my group teamed up with the Levin lab in 2019, to ask the following question. Could you ask an evolutionary algorithm to also rearrange frog parts early in development to deflect development away from the default phenotype, which is a healthy, perfectly formed Xenopus laevis frog? Could you deflect development away to discover a Xenopus adjacent organism like the one you just saw on the previous slide? We were particularly interested in how far away from the default developmental trajectory this evolutionary algorithm could deflect development. In other words, could the evolutionary algorithm guide us through xenospace, the space of all possible Xenopus adjacent organisms? The answer, it turns out, is yes. In this case, Doug Blackiston told us that he could liberate two types of cells from early frog embryo: frog skin cells, which are going to model using blue voxels that are passive and soft. And he could also liberate myocardiocytes, heart muscle cells that spontaneously increase and decrease in volume, which we're going to simulate as red-green voxels that spontaneously increase and decrease in volume. Here you see three initial random simulated xenobots taken from a population of 100 random simulated xenobots. You can see that evolution is trying out different 3D geometries, and it is also trying out different combinations of skin and heart muscle tissue. Normally, myocardiocytes in an adult frog will spontaneously synchronize and pump blood in and out of the heart. Doug told us he wasn't sure whether when we rearrange myocardiocytes in a xenobot, whether those cells will synchronize. So we told the evolutionary algorithm to be conservative and assume that they will never synchronize. What the evolutionary algorithm is trying to do here is evolve a simulated xenobot that moves from left to right as quickly as possible, but it has to build these simulated machines out of unreliable or at least unsynchronized parts. And it turns out that this is possible. This was the end result at one evolutionary trial, which results in a xenobot that moves from left to right, despite the fact that the myocardiocytes in the ventral or bottom surface are unsynchronized. So a couple of interesting observations to make about this particular xenobot. It's an interesting example of morphological computation. You can see that the soft passive skin cells on the top, the blue cubes, they're kind of absorbing all of the individual unsynchronized impulse forces coming from the myocardiocytes on the ventral surface. It's sort of averaging them or smoothing them out a little bit, which leads to a regular oscillation that leads to reliable left to right travel. Here you see Doug, the microsurgeon, building a physical xenobot. This is a snapshot partway through the manufacturing process. At this point, Doug has separated out skin and heart muscle cells from very early frog embryo. These cells early in development are sticky and they kind of glom back together in a mass. As they do so, Doug carefully layers down skin, heart muscle, like a sandwich, following the evolutionary blueprint of what evolution was able to come up with. And he's putting the finishing touches here on a four-legged xenobot that's flipped on its back. You can see Doug scraping away unwanted cellular material. At the moment, in Xenobots, we've automated the design process on the left using an evolutionary algorithm, but obviously the manufacturing process is still very manual. It takes Doug, who is an extremely talented microsurgeon, about 4 hours to make one of these Xenobots, which is about 1 millimeter across. Given that fact, you can imagine we're quite interested in trying to automate this manufacturing process. Here in the bottom part now of this video, you're going to see Doug's best attempt to create a physical copy of this evolved simulated Xenobot. You'll see that it has slightly different shape and slightly different function from the evolved solution, but we proved that we did cross the sim to real gap, at least in this point, by taking the Xenobot that you see in the top, just a different view of the Xenobot you just saw, flip it on its back, and when we put it on its back, it doesn't move at all. We lose that function. So this shows that that 3D evolved geometry dictates function, not just in the simulated Xenobot, but in reality as well. Just for fun, we put a bunch of these Xenobots together in a dish, and you get interesting collective behavior. Given the 3D shape of these Xenobots, they temporarily adhere to one another. They sort of mechanically lock together and rotate about their common center of mass before separating again and heading off in their own individual trajectories. It may be difficult to see in this video, but there are a couple of very small particles that have been placed in the bottom with the physical xenobots. You can see this more clearly in the upper half. As these xenobots move about, they unintentionally push some of these particles into piles. These are inorganic particles, but it led Mike Levin to wonder what would happen if we replaced these inanimate particles with animate particles, with biological cells. I'll come back to that in a couple of minutes. When you build robots from living tissue, you're in essence building a robot made of robots. The rich inner life of cells, I would argue, pre-trains them, in this case, to zero-shot learn external surprise. They immediately begin to heal themselves over a few hours after grievous injury. Doug cut the xenobot almost in half. They're able to do this, they're able to recover from this unexpected external surprise with the additional surprise that obviously they're in the form of a xenobot, not a default frog. Cells have had 3.5 billion years of practice of grappling with what from their perspective is external change. During development and growth and damage and regeneration, their cellular neighbors are constantly changing. From the organismal level, that is internal change, internal cellular reorganization and adaptation. And that collectively pre-trains the organism, or the xenobot in this case, to grapple and deal with external change surprisingly quickly. More recently, we've created xenobots that swim by beating very small hairs on their outer surfaces called cilia. Turns out that we can genetically manipulate these swimming bots, which is what Doug did in this experiment, so that they go, see, remember, come back, and tell. They do so by fluorescing green by default as a result of this genetic manipulation. However, if they come into contact with an environmental stimuli of interest, which in this case is the blue light, they will switch from fluorescing green to permanently fluorescing red. If we then collect from the field back our xenobots and we count up the ratio of red to green xenobots, we get a rough estimate of how many of them came into contact with this environmental stimuli of interest. I wanted to focus on this particular part of this paper. I thought this would be particularly interesting to the robotics crowd here. It points to, again, a way of using these xenobots as miniature robots for perhaps inspection tasks. Most recently, we found that a xenobot, the shape of a xenobot can cause it to push loose frog cells into piles in the way that they earlier pushed inanimate particles into piles. Depending on their shape, they push cells into larger or smaller piles. Over several hours, if a pile becomes large enough, that pile will grow cilia on its outer surface and start moving on its own. In a way, this becomes a child xenobot. Interestingly, you can view this as a form of replication that, as far as we know, has not been observed in the natural world. Plants and animals reproduce via growth. They either grow seeds on their outer surface and shed them, like plants, or they grow copies of themselves internally, like mammals, and birth them. Turns out that this replication process that we observe in xenobots is actually much closer to the way von Neumann originally envisioned robots self-replicating back in the 1960s. They would collect raw materials in their environment and manipulate that raw material to build copies of themselves. Of course, this is not an autonomous process. We had to prepare the environment for these xenobots in a particular way. We had to sprinkle these dissociated frog cells into the dish. We had to provide these raw materials to the xenobots in order to replicate. But even when we do this and supply them with large numbers of dissociated cells, undesigned xenobots, those that just form spontaneously, They can produce children like the one that you see here, but these children tend to not be able to produce viable grandchildren. The children don't move very quickly or very well. They're not very good at pushing cells into other piles. So they produce grandchildren grandchild piles that are too small to grow cilia. They don't mature sufficiently, so those grandchildren never move. And so these default xenobots lose this replicative ability after just one round of replication. We showed in this paper that, again, we can come in with an evolutionary algorithm, wrap this around, in this case, simulated xenobot swarms, and evolve those swarms to postpone the loss of replicative ability for as long as possible. How does this work? Well, here you see one swarm taken from a population of swarms. We have multiple swarms at the beginning of our evolutionary run. This particular swarm, the shape of the individuals in this swarm are spherical, which is sort of how default xenobots form. If you don't surgically manipulate them, they form into a sphere of spheres. These spheres, as you can see, don't create very large piles. They do, however, in this case, create one pile, which you can see in the bottom right, in the center right of this animation, that reaches 74% of the size required to be large enough to grow cilia and start moving on its own. So that pile center right doesn't become big enough to move, so the replicative process doesn't even reach one round of replication. It was 74% of the way there, so we assigned this swarm a fitness of 0.74. This swarm, as I mentioned, is just one of several swarms in a population of swarms, and we assign fitness based on how big a pile they make and whether or not they achieve rounds of replication. A few generations later in this evolutionary process, this swarm of 9 donuts evolved. This donut shape or toroidal shape manages to build one pile that is large enough to become a child xenobot, which earns this swarm 1 fitness point. That child, in turn, creates a pile that is 51% of the way to being large enough to become a motile grandchild, which earns it another 0.51 of a fitness point. We combine those together. This particular swarm achieves a fitness of 1.51. One round of replication and 51% of the way to a second round of replication. Much later in this evolutionary trial, this swarm of Pac-Men evolved. This swarm of Pac-Men collectively build 3 children, which in turn build 1 grandchild, thereby postponing the loss of replicative ability for 3 generations. We get 3 generations in this case. Here are the end results of 48 other evolutionary trials. Some of these xenobot designs that you see here, when built as in simulation, were able to postpone replicative loss for 5 generations. And when they were built as physical swarms, some of them managed to cross the sim-to-real gap in this case. The physical swarm of xenobots was able to replicate for 5 generations before losing the replicative ability. So in essence, we've been exploring an evolutionary algorithm that can enrich a behavior that is rudimentary or latent in this genetically unmodified living material. Of course, making robots that only self-replicate is of limited practical use. As demonstrated by von Neumann back in the 1960s, if one goes ahead and bolts component D onto a replicator, where that replicator is made up of parts A, B, and C, several things become possible. First, if the description of D is in the replicator's tape, phi, then those changes are inheritable and become D prime. And if we continue this process, what machines do once replicated will begin to change. The function that D gives rise to produces a slightly different function in the replicated machine that has a D prime component. That paves the way towards evolution. The second result of this D component is that if D performs some useful function, regardless of whether D mutates over rounds of replication, as the number of self-replicating machines increases, the amount of D that's performed, the amount of function that D does increases superlinearly. You get exponential utility. You get a swarm of machines that become exponentially more useful over time. We explored whether self-replicating xenobots might actually be capable of exponential utility. We explored this just in silico. To do so, we placed simulated xenobots and loose frog cells, represented in gray here, into partially constructed circuits, shown in black and red here. If xenobots moved even at random, they could sometimes push small, flexible, adhesive wires into one another, like you see in the small two bottom insets. If these flexible wires close a circuit between a power source, represented by the red dot, and a light source, represented by the crossed circles, the light source is turned on. We treated the number of light sources switched on in a circuit as a small amount of useful work. That's the D here. In this video, you're going to see 9 xenobots turn on the light source initially in the top right. In the top left. You'll notice that they close the circuit between a battery and the light source, and the light source top left turns on. In the upper right, in a moment, they're going to, through random action, close the circuit to turn on the light at the top right. So this particular xenobot swarm, it replicated. You'll see there's some irregularly shaped pink bots in the dish. And as a side effect of replicating, they've done a little bit of useful work. They've turned on 2 of 4 light sources. Clearly, this whole system is unlikely to transfer from simulation to reality anytime soon. We simply wanted to know whether 2 emerging technologies, xenobots and flexible electronics, when combined in future, might be capable of exponential utility. Can a growing number of xenobots fix a growing number of flexible circuits? The answer, it turns out, again, in theory, is yes. If the swarm in circuit A manages to turn on at least 1 light, we pull all of the child xenobots out of A, break that group of child xenobots into 2 equally sized groups, and put half of the children into circuit B and the other half of the children into circuit C. We then sprinkle fresh frog cells into circuit B and allow those child xenobots to do their thing in those 2 circuits. By repeating this process, we can ask whether we end up with an exponentially growing number of circuits being fixed by xenobots. You see this branching tree of more and more circuits. One of these branches will come to a halt if in any one circuit, if 1 or 0 children are created. If only 1 child is created, we pull that 1 child out of the circuit. We can't break it into 2 halves. We break it into 2 groups, 1 child and 0 child. So that second circuit cannot be inoculated with any xenobots. That left-hand branch dies out. We put that single child into the right-hand circuit, and if it produces children, we keep going. If a swarm produces no children, then there are no subtrees growing from that particular circuit. The tree in panel D here helps us to answer the question of whether exponential utility occurs in this setup. At the center of this tree, you'll see there's a red line next to the red dot, and that red line next to the red dot denotes the ancestral circuit, circuit A, in which one light was switched on. So in that case, there was a small amount of utility. The gray and red lines branching off from that inner red line denote 2 other circuits, B and C, inoculated with child xenobots. So circuit A produced enough child xenobots to inoculate circuits B and C. The gray line The gray line represents the fact that no lights were turned on in circuit B. The red line indicates that in circuit C, at least one light was turned on. And you'll notice two branches branching out from each of those two lines, representing that there were children that appeared in those circuits. You'll notice in the tree as a whole that some branches die out. These represent circuits in which no child xenobots were created with which to inoculate two new circuits. If we march from the inner circle of this tree outward and count the number of red lines we cross at each concentric circle, we get the solid curve reported in panel E, showing you that although exponential utility didn't occur, we definitely got superlinear increases in utility. This technology, this swarm of replicating xenobots, became superlinearly useful over time. That concludes our tour of some of the more interesting corners of the space of all possible robots, I think. Although I've been your tour guide today, allow me to thank some of the people who found those places to begin with, including my students, Sam, Atusa, David, and Sita. I'd also like to thank my colleagues at Tufts, Mike Levin and Doug Blackiston, our colleagues at Yale and the City University of New York. And finally, I'd like to thank all of those who funded our explorations. Thanks very much, and I'm happy to take any questions.